Developing new treatments for neurodegenerative diseases is no easy task. That's because these disorders don't just affect one cell type. The central nervous system contains a complicated array of cells that rely on a precise arrangement to facilitate cell-cell communication. And this complexity is difficult to replicate in the lab. Now, scientists from 𝙎𝙏𝘼𝙍 have started to make headway on this problem. They're using so-called spinal organoids, which mimic the developing spinal cord in both form and function, to gain new insights into neurodegenerative disease. To create the organoids, they used a chemical cocktail to coax stem cells into morphing into different types of spinal cord cells. These cells can be encapsulated in a special type of gel that replicates the extracellular environment found in the spinal cord. As the cells grow, they form the motor neurons, interneurons, and astrocytes found in native tissue. The team applied the method to stem cells derived from healthy people, as well as from patients with spinal muscular atrophy, or SMA. SMA is a genetic disease that causes the degeneration of motor neurons and primarily affects children. The consequences can be severe, including paralysis and even death. The researchers found that motor neurons developed similarly in healthy and diseased cells until the organoids reached full development, around day 35. At this point, the motor neurons in the diseased organoids began to degenerate. This finding implies that SMA might not occur during nerve development as previously believed, but more research is required. Further investigation showed that the gene mutation that leads to SMA causes motor neurons, which don't normally divide, to begin dividing again, ultimately leading to their death. When the researchers used a chemical to block this division process, the neurons mostly survived. The team will continue using the method to pinpoint other cell types potentially affected in SMA and in other neurodegenerative disorders. Their hope is that the organoids will reveal new insights into disease development and offer an avenue towards preclinical drug testing.